Hi, this is going to be the notes video for meiosis that you should watch if you were absent in class last week. So really quickly, I want to review a few things. So at this point on your note sheet, you should not be writing anything down and you should just be listening. I will tell you when it is time for you to start writing. To review what a chromosome is, it's going to be a single molecule that's made of DNA and proteins. In this case, the DNA is represented on the left in blue, and the proteins called histones that DNA is wrapped around are represented in yellow. So when a cell divides, it's going to need to replicate all of that DNA, and it's going to wind it up into chromosomes. Now, that chromosome that you see over here on the right, you'll notice that there's two halves. There's this half and there's that half, and they're joined in the middle by a centromere. Chromosomes in different organisms. So each organism has a unique number of chromosomes, each species, I should say. Pea plants have 14, sunflowers 34, cats 38, so on and so forth. Humans have 23 pairs of chromosome, one coming from each parent, for a total of 46. Every single organism that's produced by sexual reproduction is going to have two copies of each chromosome, one coming from each parent. So you can think of one chromosome from maybe your dad as 1A and the other one of the same type that came from your mom as 1B, or vice versa. Now, each of these are going to replicate when it's time for cell division. So in this diagram over here, you're seeing 1A replicating and also 1B. Those two copies are called homologous pairs or homologs of one another. They are the same size, the same shape, and they carry genes for the same trait. So again, looking at this diagram, we have 1A and 1B, and now 1A is replicated and 1B is replicated over there. What is a diploid number of chromosomes? So diploid is a term we've talked about before, and what that means is you have two sets of chromosomes in your cell, one coming from your paternal parent and one coming from your maternal parent. So those would be all of your body cells, cells of your skin, of your brain, of your heart, anything in your body except for sperm and egg cells are going to be diploid. They are going to be said to have 2N because, again, they have two pairs of every single chromosome. In humans, again, we have 23 pairs, two of each one is going to give you that 46. Sperm and egg cells, though, are called haploid because they have half of the normal diploid number. So they're said to be 1N, which you see down here. They only have one of each chromosome for a total of 23. Here's where you need to start writing things down. There's a difference between reproduction that happens asexually and reproduction that happens sexually. So asexual reproduction is the production of offspring from one parent. So mitosis, which we had talked about before, is an example of asexual reproduction. There are other types of asexual reproduction that we've also talked about. Binary fission is how bacteria replicate themselves. Vegetative propagation is something that plants do, so on and so forth. There's a bunch of different examples, and I'm not going to list them all. But asexual reproduction is going to result in offspring that is exactly the same as the parent, barring any mutations. Sexual reproduction, on the other hand, is done through the fusion of two gametes or two sex cells, and that is going to result in a lot of genetic variation. So meiosis is the process of getting cells prepared for that process of sexual reproduction. So meiosis is really the sexual reproduction division of that nucleus. And so the result there at the end of meiosis is going to be 4 cells that have half of the number of chromosomes that a normal diploid cell would have. The creation of gametes or sex cells, again, are genetically different from the parent cell, which would make sense, right? Because if you have two regular diploid body cells and you fuse them together, then you suddenly have twice the number of chromosomes that humans are supposed to have, and having too many or too little chromosomes is a problem for several reasons in offspring. Much of the time, genetic discrepancies, chromosomal abnormalities like that result in non-viable offspring that won't even survive. So that's why we have to split the genetic information in half to recombine it to get a cell or a zygote with a normal amount of genetic material in it. So what exactly happens there in meiosis? It's only going to occur in cells of the sex organs. So in males, it's going to occur in cells in the testes, and in females, it's going to occur in the ovaries. That process in males is referred to as spermatogenesis, and it's going to produce 4 functional haploid sperm cells that you can see down at the bottom of that diagram on the right. In females, that process is called oogenesis, not oogenesis, but oogenesis. And an interesting thing happens in oogenesis, and that is that the cytoplasm divides unevenly, and it's going to produce 1 functional haploid egg, which you can see over here. And it's also going to produce 3 polar bodies, which you can kind of see over here. And those polar bodies don't have enough cytoplasm in them to be viable to do anything, so they just get reabsorbed. There is a big size differential between sperm cells and egg cells. This diagram in the middle kind of misrepresents the differential in size. The 2 actual cell images are a little bit better at representing that size discrepancy, because sperm cells, their only job is to swim really fast and fertilize an egg. The egg, once it's fertilized, has to mature into an actual baby. So it makes more sense for the egg to be much, much bigger than the sperm, because the sperm is essentially just a little shuttle car to take genetic material to the egg. So once again, meiosis produces cells with half the number of original chromosomes. And how that works is the DNA is going to replicate once at the very beginning of meiosis, and then the nucleus is actually going to divide twice. So benefits to mitosis, benefits to sexual reproduction are really all about genetic diversity. And genetic variation among a species, because if you have more genetic diversity in a species, that species is more likely to survive long term, because any disaster event would be less likely to kill off the entire species, and so that species would survive overall. And one of the things that actually contributes to genetic diversity during the process of meiosis is something called recombination, where crossing over events are going to occur between chromosomes during that process. So you see that represented over here on the right. You'll see these 2 kind of crossing over each other. An actual exchange occurs between the 2 chromatids that are next to one another. So you see over here and over here, these chromatids now are combinations of both. They've exchanged information with one another. We're not going to watch this video right now. The other thing that contributes to genetic diversity is the law of independent assortment. And sometimes that's a little difficult to wrap your brain around. But remember the 1A and 1B chromosomes for a second. So let's say we have a 1A chromosome and a 1B chromosome. I'm going to make them 2 different colors. They are going to have to replicate, okay? So now we're seeing replicated, and I know this isn't a very good drawing, but when the cell divides, and remember, in meiosis, it divides twice. What does not happen when a cell divides is all of the 1As go that way and all the 1Bs go that way. Sometimes it's random. Sometimes the 1B could go to that side and the 1A could go to this side or whatever. which is all well and good. Like, it doesn't, there's no rule stating which direction chromosomes are going to go when they split apart. But during that second division, these two held together by that centromere are also going to have to split. And once again, the ones on the left don't always go this way, and the ones on the right don't always go that way. Sometimes the one on the right goes over here, and the one on the left goes over there. And that seems a little bit arbitrary. But when you're talking about 23 pairs, 2A, 2B, 3A, 3B, 4A, 4B, et cetera, with multiple divisions, the chances of having the same exact setup for each chromosome become infinitesimally smaller, okay? So that random sorting of chromosomes yields a lot of diversity in the sex cells. So basically, the law of segregation is another thing that can happen, which I just described in the previous slide. So the law of independent assortment and the law of segregation are very, very similar to one another. And they're talking about those chromatids that are bonded by the centromere. They can sort independently of each other as well. So there's no, again, there's no rule stating that all of one side is going to go to the left and all of the other one is going to go to the right. The important part about that is that human gametes then, per parent, there's over 8 million possible combinations of those 23 chromosome pairs. But then if you consider the fact that there's not one parent involved, there's two, then you really have to consider that there's 64 trillion unique combinations in a particular zygote, because there's the 8 million from combinations possible from the maternal parent, and there's the 8 million possible combinations from the paternal parent. If you multiply that together, that's over 64 trillion unique possibilities per kid. So what you will never see is, you'll never see one sibling and another sibling that look genetically identical to one another unless they are identical twins. And that's something that I'll talk about a little bit more going forward. Stages of meiosis. There are two phases overall of meiosis. There's meiosis 1 and meiosis 2. Remember, because there's two divisions that occur. In prophase 1, it's going to look very, very similar to prophase of mitosis. The chromosomes are going to pair up to form a tetrad, though. And tetrad really just means group of 4. So you can see down here in the diagram, these are your tetrad for, let's say, chromosome 1, and this would be your tetrad for, let's say, chromosome 2. And the blue one is going to represent 1A and 2A, and then the green one would represent 2B and 1B. All right. So then after the normal stuff that also happens in prophase, like the nucleus disappearing and the spindle fibers starting to form up, then in metaphase 1, those homologous chromosomes are going to line up in the middle of the cell, just like in mitosis, except you're seeing that tetrad still occurring there. Then in anaphase 1, those homologous pairs are going to separate and pull to opposite poles. So again, you notice that all the blues didn't go one direction and all the greens didn't go in another. It was random. Then in telophase 1, you're going to get that nuclear membrane reforming. The cell is going to have 2 nuclei. Cytokinesis is also going to occur, dividing those 2 cells. Now, in meiosis 2, the DNA is not going to replicate again. It's just going to divide, and those sister chromatids will eventually separate here. In metaphase, again, those are going to line up in the middle. In anaphase, the sister chromatids are going to be pulled apart by the spindle fibers. In telophase 2, the nuclear membrane is going to form around those chromosomes. And finally, cytokinesis is going to actually split the cells apart. And you're going to get those 4 reproductive sex cells, sperm cells in the case of men, or the 1 functional egg cell in the case of women. And just really quickly before I stop, here's a nice comparison of mitosis and meiosis side by side. This is not the full process, but what I'd like to draw your attention to is that you start out with 1 cell for both processes, but at the end of mitosis, you end up with 2 cells that are genetically identical, to the first cell, but in meiosis, you end up with 4 cells that are genetically different from the first cell. Okay, that's all I have for you for this particular notes session. So come back and watch the genetics notes if you are absent in class next week.